Welcome back to the session on nucleic acids. We'll continue with the presentation. Um, so, as we have already mentioned, nucleic acids, they are polymers of nucleotides, and a single nucleotide, that is a monomer of the nucleic acid, a single nucleotide, if we look into, it is composed of three molecules, nitrogen. It is a pentose sugar to which the nitrogenous base as well as the phosphoric acid is attached to. To the first carbon of the pentose sugar is attached a nitrogenous base, and the fifth carbon of the pentose sugar is attached to the phosphoric acid. Now looking into the structure of nitrogenous base, as we have already seen, nitrogenous base, they are heterocyclic nitrogen containing organic compounds, and there are two kinds of nitrogenous bases, purines and pyrimidines. Looking into the structure of pyrimidines, they are monocyclic molecules, and they have a benzene type ring, that is 6-membered ring, and since it is a heterocyclic compound, they have nitrogen on the first and the third position. Okay, and there are three kinds of pyrimidines, that is cytosine, uracil, and thymine. Cytosine, they do have amine group attached to the fourth position and a keto group at the second position. So it is 4-amino pyrimidine 2-on. Uracil, which is found only in the RNAs, they have two keto groups at second and the fourth position, and hence the name pyrimidine 2,4-dione. Now coming to the thymine, it is a modification of uracil, that is, apart from these keto groups at the second and the fourth position, they have a methyl group attached to the fifth carbon atom, and hence the name 5-methyl uracil. And we have already seen that the pyrimidine, it is attached to the first carbon on the sugar, isn't it? First carbon of the sugar. And the pyrimidine, it is attached via the first nitrogen, or N1. So the N1 of pyrimidine, it is bound to or linked to the first carbon on the pentose sugar, and this bond, it is known as a glycosidic linkage. And the product which is formed by the linkage, glycosidic linkage of the pentose sugar with the pyrimidine, it is known as a pyrimidine nucleoside. Okay. Now coming to the purine. Purine, it is a dicyclic molecule which is formed of fused 6-membered ring and a 5-membered ring. The 6-membered ring, it is benzenoid in nature, while the 5-membered ring is imidazole ring. You can see the numbering. In the case of the pyrimidine, it is you can see here, it is clockwise, starting from the basal nitrogen, it goes like this: 2, the third nitrogen, 4, 5, 6. Okay, and you can see here the basic pyrimidine structure is having alternate single double bonds. Okay, now coming over here to the purine, purine is having numbering clockwise. Of course, the first number, it is on the nitrogen, and then it goes clockwise down 1, 2, 3, 4, 5, 6, and then 7, 8, 9. Okay, now there are two kinds of purines, adenine and guanine. Adenine, it is having an amino group on the 6th position of the purine, so it is 6-amino purine. Okay, now guanine, it is having a keto group at the 6th position, while amine group attached to the second position. So it is actually 2-amino purine 6-on. Now how the purine is attached to the pentose sugar? Purine, it is attached to the pentose sugar via the nitrogen. Okay, nitrogen at the 9th position. So it is through this. Unlike the first nitrogen bond forming a glycosidic bond in the case of pyrimidine, here it is the N9 of the purine. It binds with the first carbon on the pentose sugar. The bonding, it is glycosidic linkage. Okay, so I hope this is clear. Now the next is pentose sugar. So we have seen the numbering of the nitrogenous bases is 1, 2, 3, 4, 5, 6, 7, 8, 9, isn't it? But in the case of pentose sugar, it is 1', 2', 3', 4', and 5'. Why the numbering like this? To differentiate it from the number on the nitrogen. So if we say it is 5 carbon, it is 5 carbon on 5th carbon on the nitrogenous base. If we say it is 5' carbon, it is the 5' carbon of the pentose sugar. Okay, so just by denoting the number itself, we can very well understand which compound, whether it is pentose sugar we are speaking about, or whether it is the what you call nitrogenous base we are speaking about. Okay, so we have a numbering 1', 2', 3', 5', and 4' and 5'. Okay, so the structure which you are already familiar with when we have studied the carbohydrate, the monosaccharide structure. Okay, so this is the structure. We have what you call a pentose, a 5-membered ring with an oxygen in the ring. We have the 1, 2, 3, 4, and 5 carbon atoms. The 5th carbon atom in the form of CH2OH. And in the case of ribose, we can see the 2nd carbon is having an OH group attached to it, while in the case of 2-deoxyribose, so these are two are D sugars. We are already familiar that there are two kinds of sugars, D sugars and L sugars, isn't it? So here it is a D sugar, and the second position, unlike the ribose, in the case of deoxyribose, they have only a single hydrogen atom. Okay, so yeah, so ribose and deoxyribose in the case of RNA and DNA, it exists in cyclic form. Okay, so we can see here. So the second position hydroxyl group here you can see, and what happens to the deoxyribose? So it is 2-deoxyribose. This is the structure. Okay, and this structure, it is known as a furanose structure, and hence the name is beta D ribofuranose. Why it is D? Because the penultimate carbon is having hydroxyl group on the right side. Okay, that is why it is known as a D-ribose. When it is cyclized, what happens? It becomes D-ribofuranose. Now, why beta? Because the hydroxyl group is on the upper side of the ring. Okay, so same here. Since it is deoxy, we have just modified the name beta D 2-deoxyribofuranose. Okay, so this is the molecular formula C5H10O5, the very common monosaccharide having the formula CnH2NON. With 5 carbon, it will be C5H10O5. But coming to deoxyribose, that is, it is 1 oxygen less than ribose, so it is C5H10O4. Okay, so how the nucleoside is formed? We have already seen that when we mentioned about the pyrimidine and purine, how it is bound to the pentose sugar. We saw that the pentose sugar, it is attached to the nitrogenous base, and the compound formed is what is referred as a nucleoside. Okay, this pentose sugar, it could be ribose or deoxyribose. On basis, it could be purine or pyrimidine. And when it is a purine base forming the nucleoside, we call it as a purine nucleoside. And if it is a pyrimidine, we call it as a pyrimidine nucleoside. Okay, now, so how we have already mentioned it. So the base, the nitrogenous base, it is joined to the 1' carbon of the pentose sugar by way of the glycosidic bond. Okay, when we refer to the glycosidic bond for the first time when we studied carbohydrates, we saw that the glycosidic bond, it is of two types based upon through which atom the glycosidic bond is formed, isn't it? So in the case of carbohydrates, it was O glycosidic bond, while here it is N glycosidic bond. Okay, here this is N glycosidic bond. Okay, because it is the nitrogen over here, which is actually forming the bond, isn't it? So it is N glycosidic bond. Okay, so it is a beta N glycosidic bond through which actually links the nitrogenous base with the pentose sugar. Okay, it is N1 to 1' carbon. I hope it is clear. And how it is formed? We saw that onto the pyrimidine, this is a pyrimidine, isn't it? Just a single ring. Onto the pyrimidine, sorry, what do you call it? Here it is, I hope you remember. Yeah, okay. So this was the structure, right? We are having NH over here. In every pyrimidine, we have a NH over here. Okay. So when this was, and what about this one? It is OH. You remember? This is OH. Okay. Here also, deoxy sugar also, it is OH. So the OH on the first carbon, 1' carbon, and hydrogen on the first nitrogen, it is released as H2O. So by removal of water, a glycosidic linkage is formed. It is a condensation product. Okay, and the product, it is actually what is known as a nucleoside. Okay, the nucleoside, as we have already mentioned, based upon which kind of nitrogenous base, we can have two kinds of nucleosides, purine nucleoside and pyrimidine nucleoside. We have already seen that pyrimidine nucleoside is formed by the glycosidic linkage between N1 and 1' carbon, right? While purine nucleoside, it is formed by glycosidic bond between N9 of the nitrogenous base with 1' carbon of the pentose sugar. I hope it is clear. Okay, now depending upon which kind of sugar is involved in the formation of nucleoside, we have two kinds of nucleosides, ribonucleosides and deoxyribonucleosides. Okay, and each of these nucleosides can be further divided into two, that is based upon what kind of base comes into play. If it is purine, we have purine nucleosides, and if it is pyrimidine, pyrimidine nucleosides. Okay, purine nucleosides, they are like adenine and guanine nucleosides. They occur in both what you call RNA and DNA. Okay, and these are known as adenosine and guanosine in the case of RNAs. And if it is a deoxyribose sugar forming the binding with the adenine, we call it as deoxyadenosine, and if it is guanine binding to deoxyribose sugar, we call it as a deoxyguanosine. Okay, and pyrimidine nucleosides, three types are there, isn't it? Cytosine nucleosides, they occur in both the DNA and RNA. Unlike the naming in the case of purine nucleoside, here you can see it is cytidine. Okay, since cytosine containing nucleoside is found both in RNA and DNA, to differentiate that, we have cytidine in RNA and deoxycytidine in DNA. Uridine, we have only one. It is found only in RNA, so we don't need a deoxy form. We don't have a deoxy form. So it is uridine in RNA. And similarly, thymidine, we don't need a deoxy. Thymidine is always in deoxy condition, isn't it? So it is thymidine, means it is in DNA. Okay, so I hope the nucleosides, the naming is proper with. Okay, you can understand. Now, ribonucleosides, you can see here ribose sugar. Okay, this is a what you call adenine group. So it is adenine bound to ribose sugar. It is adenosine. Okay, this is cytosine bound to ribose sugar. Since we have only one form of cytosine containing this one, it is the cytidine. Okay, ribose sugar, it is cytidine. Okay, then guanosine, that is guanine attached to ribose sugar. Uridine, we have only one kind of uridine. Okay, we don't have deoxy. All others we have deoxy as well. Okay, we have deoxyadenosine, we have deoxycytidine, we have deoxyguanosine. If instead of this ribose sugar, we have deoxyribose. Okay, uridine, we don't have deoxy form. Always the uracil, it gets attached to only ribose sugar. Okay, now, I hope that part is clear. Okay, so we have ribonucleosides as well as the deoxyribonucleosides. Okay, now nucleotides we will look into in detail in the coming presentation. Okay, thank you.